and good morning, everybody for listening. And the topic of our presentation will be Bayesian change point segmentation and genome annotation. And we will have two presentations now on this topic. One is mine, and the second is of my master students who are entering now graduate school, who made a really brilliant implementation of the segmentation approach to the chromosome duplication biases. So I think I consider my own presentation as a kind of a popular introduction to the theme, and then I leave the field to Sasha and Sergey. And first of all, I want to say a couple of words about what is genome annotation. And currently, genome annotation usually is considered as a kind of a partition of a genome into the kind of a segments. Some segments can be just over the length of one letter. And those segments represent local functional information. Those who've seen BET files, and so this is the basic engine under the hood of a genome browser, Santa Cruz genome browser, for instance. They have seen them as a kind of a standard format with a left boundary, right boundary, and some functional description of the segment. So this could be, for instance, protein coding, non-coding regions, exons, introns, and maybe intergenic regions, or some regions of persistent histone modification, maybe some regions of persistent DNA methylation, and also levels of phylogenetics conservation, conserved region, and plethora of other features. The point is that the competing approach is to make some coloring, maybe some not segmentation, but function with the genome coordinates and arguments. So in this case, you have some numbers like frequency of methylation degree, or maybe degree of conservation, or maybe again frequency of histone modification. So in this case, we have some kind of smooth curves around the DNA. But very often those smooth curves are flattened to some segments with a high and low level of something. And I will talk in my presentation about this kind of a approach which can do this more or less automatically. Also, from the functional point of view, if you are annotating a genome, you can use some kind of a direct functional annotation. For instance, the most simple example is a protein coding regions. You can do some kind of reverse mapping of amino acids. Now you can map transcriptome to a genome. So you're using direct experimental information on the particular segments of DNA, how they involved into production of RNA, for instance, if you map transcriptome. Also, you can use some kind of indirect data. For instance, you know that there are important segments and regulations like CPG islands, and you know that those sequences are involved into the CG-rich promoters. They are methylated. You know nothing about methylation pattern, but you know that usually they are CG-rich with the G and C letters. So and also they're rich with CG pairs. So you can do some kind of statistical modeling and extract them out of the sequence, and also you can do some kind of algebraic sequence analysis like counting words and windows. In this case, what you observe are some statistical properties of the sequence itself, and you make indirect inference about what function properties can this correspond to. So you say that there are a lot of C's and G's, and so it's basically very likely to be CPG-rich island, and this island is kind of likely to be involved into the transcription activity regulation. So it's kind of a, and what we are going to talk about usually, that's second type of genome annotation. So we are going to talk about how to make kind of a refined mathematical conclusions out of the noisy biological data, and we have some kind of a general idea how the ideal case might be, and so there are some kind of a noise, maybe in the sequence itself, or maybe in the like results of some library sequencing, which shows you that what's you observe. So we're going to talk about basically statistical modeling of sequencing. For instance, here, this is the genome of phage lambda, and you can see that there are different windows in those genomes. So you've got, so on the x-axis is the position of the within the phage sequence, and along the y-axis, that's average frequency of G or C in this, in some window. And so you move the window and count the percentage of C and G, and here it's about 50 something, so about kind of slightly below 60%, and down drops. So this window contains very rich sequences. So here it's about 30% of sequences only C and G, so 70% is A and T, and it goes up and it goes down and so on. So the whole thing can be split into segments which are different percentage of C and G there, and in fact, this is the answer is that different genes in this lambda phage, they have different amino acid composition depending on their function in the protein of the protein they code for. And so this moving average domains in the compositional sequence, they actually correspond to different proteins which are coded into the sequence, in the sequence of the lambda phage. So those windows are, in fact, they've got some indirect functional consequences. So we can make direct inference about the functional consequences very often out of a very simple observations like percentage of particular letters in the DNA alphabet in the text. So what we are doing now, we are trying to generalize, this kind of a noisy pattern into the several segments, and between them there are boundaries. So in this science, they also called change points. So we've got decomposition of the some smooth function into a number of models, and in fact, in which domain, in each domain, the sequence is considered to be random. So it's a realization of some random process with a single parameter, and this parameter corresponds to the frequency of C or G in this domain. So there are many methods to do that, and now I go a little bit into those methods. Just a moment. So now we consider this from the mathematical point of view. Suppose that we've got a data series V, and at each point we've got some observation. It may be taken from discrete or maybe some kind of natural numbers like here. So this is the this window corresponds to the number of reads coming from some sequencing, and I think this is the inaccessibility pattern. So the inaccessibility profiles, and those places are less accessible, and those places are more accessible in terms of reads coming from the cuts DNAs may make. And and the model is, as I told you already, so the data are partitioned into a series of non-intersecting segments, and segments, each segment is considered to be a realization of independent random process, and parameters are persistent within each segment and abruptly changed at the boundaries of the segment. So that's why this problem is called change point identification problem. So we are interested in the positions of those boundaries of change points. And so we are interested in identifying the number of the segment K in the whole segments, and also the statistical properties of those segments and the positions of the boundaries between those segments. And sometimes in some formulations, we assume some of those values to be known. And this gives different approaches. And in fact, in machine learning, those approaches are considered as a kind of classes of the problem. So this is a pretty old-fashioned approach to machine learning. Now it's mostly neural networks and data-rich environment. You get a lot of data to train the neural network. And this is the old-fashioned type of machine learning where we had a pretty poor data sets, and so the people try to make inference from short sequences and trying to make some kind of optimal understanding of what's going on there out of the pretty data poor problems. So returning back to the segmentation problem, so we've got those segments. The parameter here is the percentage of C plus G. So basically, if there is a Bernoulli model, that's Bernoulli parameters, I like 57 here will be the probability of success of coming with COG here. And so at this, so the model is persistent throughout the segment, and here it's changed to another model, which is again from the same class, the Bernoulli model of generating sequence, but with another value of the parameter of the Bernoulli parameter. So here it's again the same type of the model, but other parameters play values, and those parameters are changed abruptly at the boundaries. So that's it, how it's happening. And so we don't know sometimes how many segments we should take into consideration. For instance, you may say that there should be very small, very low segment somewhere here with a very high probabilities of obtaining AOT. Or maybe you should include two segments here, or maybe you should merge these three segments into the single one. So the number of segments in this partition is important parameters, and a lot depends on it. Also, you can fix the number of classes. You can say that you've got on the two classes of sequences. That's a usual case in the studying, for instance, CPG island identification. You've got CPG rich and CPG poor sequences, and you can fix the number of classes of segments. So here you can have pretty any probability of any frequency of G plus C, but you can say that there are only two types, and one is GC rich and another one is GC poor, and no more than two. Okay, so these are different problem formulation. They depend on the parameters we assume to be given at the start of optimization. So, for instance, one of the classic formulations is a hidden Markov model. We know that segments are from one of some usually small number of classes. And usually you don't know the particular parameters of segments in the class, but the number of classes itself is given. And this is the usual case in this type of problems, because usually you don't fix the particular values of parameters, but you infer. And in this case, you have a number of classes, and you infer for the number of segments k, the positions of segment boundaries, and also for the parameters of probabilistic models within each sequence. So you have to also infer for the probability of letters in each class. Classic example of a hidden Markov model is problem of two unfair coins. You've got two coins biased in a different way, and you toss them. You toss one coin several times, and then at random point you change the coin, you switch them, and then you start to toss another one. And so the H and T's are for heads and tails, and here you've got a lot of heads, so the coin is biased into the direction of heads. Heads are more probable. You tests, random trials, and probably the coin was changed, and all the second part of the series have much more tails than heads. So in this case, you switched the coin here, and you used this coin for this part of the sequence, and another one for this part of the sequence. So we change it somewhere here. So you have to optimize for the degree of the bias, which shows you how much more probable to have H than T. And also you optimize for the position of the boundary, and also you optimize for number of segments, so how many times the coin was changed, switched. But you know that there are only two coins there, so this is the hidden Markov model environment. You know how many classes there in the sequence, there in the problem. That's a kind of a graphic representation. So there are three outcomes here and on this graph, because I just stolen it from the book. And so there are three observable outcomes. It's important that, and x1 and x2, they are classes of the random processes. They're called hidden states. That's because we don't observe the coin. We observed only heads and tails. We don't know what the coin is. So this is the hidden state, the coin which we are experimenting with. So we've got those hidden states here. So one coin and second coin, we can use the same coin in the next trial. We can switch the coin. Then we can use the second coin for the same trial. Second trial, we can switch the coin. And what's important in the HMM environment is that you can have all outcomes from all classes you've got. So in both cases, you can have heads and tails. So you cannot distinguish between hidden states using observable states only. For any particular experiment, you don't know which actually hidden state was there to obtain your outcome. But using statistical parameter of these observations, series of observations, and using some kind of optimization algorithm, you can optimize using usually the algorithm is called the table approach. You can optimize for all those quantities we need for. Another approach is that we know the exact number of segments. Again, we don't know the statistical parameters of the segments. We don't know the positions of the boundaries, but we know that there are exactly 4 segments in the sequence. And we have to optimize for those parameters for boundaries positions, and you can infer segment type, which is the same as the parameter of the segment. And I'm not going to delve into the algorithm. That's so-called Fogel-Lorentz segmentation. It's a dynamic programming algorithm. The basic idea is that you calculate some objective function for the quality of segmentation of the sequence into 2 segments, putting boundaries in any possible position between the ends of the sequence. And then you store these results, and then you consider the third cut. And in this case, you can use the results obtained in the previous round, because one of the cuts is contains the same exactly the same segment, and as wasn't the case when there were 2 segments in the segmentation only. This is a pretty slow algorithm, and it's cubic in nature. It works with the efficiency of n squared, where n is the length of the sequence and k is the number of segments. So it's very slow when the number of segments is small. When the number of segments is large. So when you are going to segment into the large number of segments or unknown number of segments, you have to try all possible segment number. You're using another approach, which is called change point dynamic programming change point segmentation. This is the model where I'm going to discuss in this presentation. So each segment has a random sequence again. So it's not as in the approaches when you count something. You consider the random models. Segments are statistically independent. It's kind of a restriction because, for instance, in hidden Markov model, you can easily introduce statistical dependence of segments. That's why it's Markov model. You can consider them as a Markov chain. Structure of the random model is the same for all segments. So the models are from the same parametric family, as I told, with the parameters of Bernoulli child, for instance. Parameters of the random models are persistent within each segment and changed only at change points. And the number of possible segment type is not fixed. So in HMM, we know the number of classes. In Fogel-Lorentz, we know the number of segments. And here we don't know the number of classes, neither the number of classes nor the number of segments. So it can be any, and that's the subject of optimization we're going to consider now. So questions: how many segments are there? Where are the boundaries? And what are the segments' properties? And this formulation can be adapted to a large inventory of problems of different biological sequence analysis. It's a very smooth way of doing things. It's easy to calculate the total number of segmentations, because in each position they can be either present or absent a single boundary. We've got 2 power n minus 1. This is exactly the number of possible boundaries within 2 ends of the sequence. So this is the number of all possible segmentations, but you can also calculate the number of possible segmentation to the case segment. Again, you don't know which of the boundary you can shuffle boundaries within each segments which are absent there, and also you can shuffle the segment boundaries when you've got a single segmentation. So if you go into this and do some arithmetic, you found that it's kind of binomial coefficient of n-1 to k-1. So but there are many of them in both cases, and so we cannot test them. Even if we got an objective function, we cannot test them. After that, we can think how to do this finding this optimal segmentation in a more or less efficient way. Also about the real numbers in real problems. For instance, chromosomes, they contain from millions to billion base pairs. Human chromosome, the largest one, it's hundreds of millions, about 100 something megabase, so it's 10 power 8. And square is too slow for that, and also it requires a lot of memory in this case. For genomic elements like enhancers or maybe RNA transcripts, CPG islands, regions of open chromatin, they're usually of thousands or maybe tens of thousands base pairs. It's possible to use exact algorithm in this case. So we are going to use an efficient algorithm, which is called dynamic programming. It was published several times. I think most early publication was in Yao in 1984. Then it was published by Royberg and Finkelstein in 1993. And I'm kind of a student, not exactly a student, but I knew as a younger scientist, Michael Royberg, really great guy and very good in teaching people in algorithms. And also later it was rediscovered several times too. And so we are going to discuss the dynamic programming algorithm, which works in square efficiency. So idea is that you segment not the long sequence of the length n, but you segment shorter sequences, and you start from very short sequences, from sequence of length 1, from sequence of length 2, from sequence of length 3, and so on. So you're going from left to right. Suppose you've got the independent of segments and independent segments, and the only thing which is required is that the function which makes the quality of a total segmentation will be additive. So if you've got two segments like here, the total objective function of this will be sum of this one segment made of 1 and another segment made of 2 and 3. If this is the property of the objective function, which is a kind of an important property, in which, in this case, dynamic programming work. It doesn't work with other objective functions. And we're trying to maximize the objective function with this property over all possible segmentations. So how we do this? For instance, we want to segment of the length 4. We've got best segmentations of 1, of 2, and of 3, and we can have 4 different options. We've got the whole segment of the length 4 without any boundaries. Then we can put a boundary here. We've got left segments intact, and we know the optimal segmentation, which is trivial, of single letter V1 from the previous round. Then for the boundary in the middle. Again, we've got the tail intact, and we can go up and look for the optimal segmentation for the two-letter sequence. In this case, we've got only two options. One will be with the boundary inside, and another one will be without. So we remember that the optimal segmentation into two letters was calculated in the previous round. And so we use this, remembering from the previous round, in this next round, so we can obtain the value of the objective function for that thing. And again, for four-letter sequence, we've got the tail intact, single letter here. And we've got the optimal segmentation for three letters, which we calculated in the previous round. And since we know that the objective function of the single segmentation is the sum of two parts of the tail and of the head, which we have calculated, we'll calculate all the shorter parts in the previous stages of algorithm, we can obtain the optimal segmentation of the four letters, knowing optimal segmentations from 1, for 2, and 3. And after that, we store all possible, we add the optimal segmentation of the sequence into four letters here. And we go to, when we go to 5, again, we add the intact tail from right, and we pick up the best segmentation to the shortest sequence from the left. And if you calculate the number of operations which is needed to come after this kind of a increasing the length of the sequence, you found that will be n square. It's even visible here. You can count the number of operations, how many comparisons and adding you have to make. Now a little bit about the scoring functions. Usually scoring function corresponds to the specific problem type. And each statistics of statistical model of the segment, it requires finding the appropriate scoring function. So it must be some kind of extremal. So it must be, for instance, the attain maximum for the segmentation we considered the best, giving homogeneous segments, for instance, and as homogeneous as possible. And also it must have this kind of a property of if you've got a boundary splitting a segment into two, then the value of the objective function for the whole thing must be as a sum of two parts. So there are different scoring functions for different approaches in this thing. We will consider three different examples. One is the compositional segmentation. This was the first example we looked at about CPG frequency within the window. Then it's correspond to Bernoulli statistics. Then we've got chromatin accessibility, the number of reads taken from each particular point. Poisson model, and also we are going to consider the problem of considering mapping bias and chromosome duplications, and in this case we've got symmetrized binomial function.
Yeah. May I ask a question?
Yes.
The fact that he chose these statistics, the Bernoulli, the Poisson, and all that, is that out of a theory or out of empirical data?
It depends on the So we can work with the Bernoulli and Poisson because it's well, it's simple random models, and they somehow they model the biological processes which are in the which underline the thing. For instance, in the case of chromatin accessibility, you've got the number of cells in which you observe the DNA is cut. You've got some particular nucleotide position, and you've got the highly diluted DNAs, and it makes cuts in some cells, in some instances of DNA, and it doesn't make this cut in other cells. So you then merge link adapters and sequence the whole library. And you've got different distribution of cuts. And in this case, there will be some kind of a rare case. What is Poisson? Is a large number of rare cases, which is exactly the same. You've got rare events of a cut of particular DNA site, yeah? And in this case, you've got the number of rare events at the same place, which is exactly the Poisson statistics. So for the single position, yeah? Mm-hmm. In the case of sequence composition, you've got the window in which you've got, say, 40 A's and 60 T's shuffled somehow. And you try to model your sequence as a windows with different composition parameter. So again, you consider within the window a kind of a series of random trials with a particular parameter, yeah? And in this case, you've got exactly Bernoulli statistics or polynomial statistics, if you've got alphabet of 4. So you have to choose this model out of the idea of the biological data you're working with from the one side. And on the other hand, you have to select simple models so that you can have your machinery of making your scoring functions more or less easy. And so you can work to the formula you're going to program and work with, you know, in real stuff with the large biological data. So basically, it's kind of, you know, it's not a theory. It's kind of a, you know, common sense in some sense, you know.
So basically, if I were to have my own task and choose my own scoring function, I would try to map it also to one of these common sense models, trying to find some simple enough distribution that I can take advantage of.
Yeah, that's true. Yeah, that's true. It must be simple enough, and also, you know, usually there are some inventory of distributions and different, you know, problem and probability theory, which more or less suitable, as I show you for the case of DNA cuts and DNA's accessibility. And also, we're using Bayesian statistics. I will go later into that. And in this case, some, you know, distributions have nice properties in terms of, you know, working with simple price and so on. So yes, exactly. I think you've got some simple distributions which can help you to obtain a closed formula you are easy to handle.
Okay. Thank you very much.
Okay, so I've got not so much time, by the way. So compositional segmentation. So we have this kind of a composition. We've got this segment. So here the segment, and it's made of several T's, theta's, and theta's are probabilities of having this letter appear within this particular segment. For instance, here we also have sequence S. S something, S1 is the letter in the position 1. L is the alphabet. So we've got, in this case, in the case of DNA, 4-letter alphabet. Also, we have within each segment, we've got the number of counts. So this, in this case of the segment 1, we've got count for A is 1, count for T, A, B, C, count for C is 4, count for G is 2, and count for T is 1. So we've got counts which are n ls, so the number of ls different is the same as the number of letters in the alphabet. And so Bernoulli random model, or maybe polynomial model, also known as, letters are obtained independently with fixed probabilities, and the sum of, so those are probabilities of having different letters, and the sum of probabilities is those probabilities are sum up to 1. So this is normalization condition. And what we are going to use, we are going to use the maximal likelihood approach to obtain the optimal segmentation. And we will consider from the, we'll start from the likelihood from the single segment. And in the case of a single segment, what is likelihood? It's a probability of obtaining a segment considered as a function of those status of the probabilities of the letters. So you've got this segment, first size segment, you just calculate the probability of having exactly the sequence. So you've got first theta here for C, then you multiply it for the next probability for A, then you multiply it for probability for T. We consider as the series of independent random trials with this underlying probabilities here. So the likelihood of obtaining some particular segment will be just the product of thetas for the letters in those in the segment. And we can also collect variable for each particular letter, and you've got this power thetas multiplied with the power of the numbers of each particular letter in the segment. So the real statistics depends not on the letters, but on the counts. That's called sufficient statistics, I suppose. That's the number of counts here. So the arguments of the likelihood are the model parameters, those quarters of variables, and the parameters of the likelihood function are counts. So we, for this particular segment, we fix the counts which are considered to be given, inferred from the segment, and we vary the model parameters here and we maximize the thing. And since the segments are considered to be independent, we simply multiply the likelihood for each particular segment to make the likelihood of the total sequence. So in the case of those quarters of counts, which is the arguments of this likelihood function, those guys live on this triangle in the three-dimensional space because you've got one condition, and the condition is that you've got the sum of them equal to 1. So if they were, and it's for 3 letters. That's for 3 letters, that's triangle. For 4 letters, you've got 4-dimensional space and the kind of a tetrahedra simplex in the middle of it. So you've got one dimension eaten by this normalization condition, and then you've got the equal distribution of all possible. But you don't think about distribution yet. You don't think about distribution, don't know the density, but the argument is that you've got all the points. In the 3-dimensional case, there were points in the triangle. In the 4-dimensional case, there were points in the simplex. All the points are live in the same, in a figure like this. So you consider distribution in this space, and you introduce the distribution density. And in this case, you can, yeah, it's maximal likelihood. It's just, it's previous slide. So you, for the whole sequence, for the segment, you've got this kind of a likelihood, which is the product in the power of the counts. And for the total segment, we've got this product of likelihoods. And if you make the logarithmic transform for that, you come up with a sum of logarithms of likelihood for segments, and which is very nice, because this is exactly the requirement we thought about when we considered dynamic programming. As far as you remember, we thought that dynamic programming works if the objective function works for the segmentation is calculated as a sum of objective functions for individual segments. And this is exactly the same condition, so you should consider logarithm of this kind of likelihood is the objective function. It will allow to work with dynamic programming. This likelihood lives in this space of, and now what we are going to do is we're going to introduce another distribution density already for those status. And this is the framework of Bayesian statistics. And Bayesian statistics exactly considers the distributions over parameter space. But we are not going to do this standard Bayesian statistics trick of calculating priors and posteriors, but we simply marginalize over some prior distribution and consider this marginal likelihood. So it's basically we start from some prior distribution, and we can in this case take non-informative prior just homogeneous distribution over all this green space, and consider this kind of integral of the values of likelihood of all possible prior values as a objective function. We can prove that this one works in this case of Bernoulli sequence. And what is nice with this thing is that this integral is taken exactly. This is so-called Dirichlet prior, and this is Dirichlet distribution, if you know about that. And this will be the closed formula for that in the case of the non-informative prior rho theta. So, and this formula contains only counts, because this is sufficient statistics for the case of Bernoulli sequence. And K, so those the counts for the segment K, and then case here is as simply as the sum of those counts for particular segment. And for the sequence, we'll get the product of this segment priors. And then we can use our dynamic programming to find the optimal marginal likelihood segmentation. And here is an example of this optimal segmentation. You can see that it's indeed gives you some spans of letters without A, for instance, or maybe two G's here. Maybe here there is a very long segments without only one A, I think, no A. So there are, but in fact, segments are too short. And there are too many boundaries that tend to sometimes select a single letter. So it's not a good example of segmentation. That's example of overfitting. So the system tries to treat statistical fluctuation as a kind of a signal, and introduces too much inhomogeneity. You must assume that there is some allowed measure of fluctuation in the sequence, which can be tolerated, and so you must introduce less segments there. So you must somehow penalize for boundaries, and this is the trick called regularization. And basically, this is the formula for the maximal likelihood for this the whole segmentation. You just add the penalty parameter beta, and you multiply for each bit for each new boundary introduced between segments. And so after you've got k segments, you've got beta power k minus 1 here. So you've got this product of likelihoods for particular segments in each particular segmentation. And again, you've got beta in the power of number of segments. So that's exactly, this happens because this two small segments, that's example of comparing in the single optimization framework models with different numbers of parameters. Because in each case, you've got 3 parameters for each segment and 1 parameter for boundary position. So segmentations with more segments contains much more parameters, and it's incorrect to optimize without this kind of any kind of a regularization models with different number of parameters in the same optimization round, because in this case always models with more parameters will win, because they have more flexibility in this case to find the optimal value. Only with the data which are very, very real structured, in this case you can by luck sometimes have a model with less parameters approximate them well. But if you introduce this kind of regularization and you select this beta appropriately, you can obtain a pretty good realistic segmentation. In practice, you've got one value from theory, and it's called a Kaiki criterion. You should logarithm beta select to be equal to the number of the alphabet. And this will be correspond of Gaussian true Bernoullian distribution with real known segments approximated with the least squared error of the number of the segment boundaries. But sometimes you don't need this, and you can use this parameter to control the number of segments. You want to have a chromosome segment in 3, 4, 5 small number of segments. You can select this beta to be very high, and in this case you've got a kind of a very rough segmentation. Just by selecting this parameter, you can control the roughness or maybe details of the results you are getting. That's what I've called by setting large penalty P. Also, because we don't have usually enough computational power for considering long segmentations, you can use kind of a divide and conquer algorithm. You can split segments into huge chunks, say by 10,000 base pairs, and overlapping by half of this chunk. Then you can segment those chunks, and then you can squash boundaries using OR rules. So there is a boundary in the merged sequence if there is any of the boundaries, and also on the ends of the sequence boundaries too. So then you can have a sequence with a much smaller number of boundaries, and then you can use a segmentation only taking into account the allowed set of the boundaries which came from the previous round. And what is important with the procedure of the dynamic programming I've showed you in the beginning, it's not important if there is a allowed boundary after each segment, or you can use a boundary, only some smaller set of allowed boundaries in the beginning. In both cases, the dynamic programming would work. So in the next round, you doing segmentation of this merged sequence, and it works surprisingly well, showing you very good approximation. Sometimes in very unrealistic cases it fails, but usually it gives you results which are very close or identical to the true optimal segmentation of the sequence. Now I'm going to, I don't have too much time now, I'm going to consider another type of scoring function. So this is Poissonian function, where sequencing some kind of a density. For instance, I suppose an example from the audience, we've got some natural numbers in each position. So this can be infinite, it can be very high. There is some expectation of this number, which is the Poisson parameter. And again, you are going to segment this data series into the segments. For instance, no way of coloring them. So here's the window with slightly more or less similar Poisson parameter. Here's another window with higher Poisson parameter. It's a window with a low Poisson parameter, window with a higher Poisson parameter, and so on. So you're going to split them into the windows with different values of the expectation of the number of reads coming from the cuts of the inaccessibility, for instance. So this is the Poissonian model. Again, you do the same trick. You using the maximum likelihood principle. In the case of Poisson model, the likelihood for the single letter, which was simply the probability of having the letter in the case of Bernoulli, will be the Poissonian distribution. So K is the observation. If you've got, here's the K. So if you've got, say, 10, you've got Poissonian parameter power 10, 10 factorial, and exponent of minus Poissonian parameter. So you can, lambda is the expectation. Sometimes people put E here as a more convenient letter for the expectation. That's for the single point. If you got segment S, you just multiply. You just multiply, and you multiply for different count. So you multiply and you multiply and you multiply, and you discover that this thing doesn't depend on the observations. So you can move it out of the product, and you've got only the number of such factors which will be exactly the length of the segment. Also, this thing in the denominator, it doesn't depend on the Poisson parameter, it depends on the counts only. And the counts, as we know, they are not the subject of the maximal likelihood, because we know them given. And also you can take it out of the product in this case. So it's only kind of a normalization factor, so it doesn't depend on the lambda, which is the argument of the likelihood function. And the final thing is the lambda will be the power of the after each next term, you will have another lambda in the power of the count, in the power of the observation, and finally I've got lambda power the sum of all the counts in the segment. So for this segment, you've got to sum up all the counts here. And this is a known thing. This is the sufficient statistics for Poisson distribution. It's known that the sufficient statistics for Poisson distribution is the sum of the counts. Exactly as is in the case with the Bernoulli, when the sufficient statistics was simply the counts themselves. So you've got the sum of the counts, and the likelihood function depends only on the length of the segment and the sum of the counts here. And in the case of Bernoulli, the length of the segment was connected. You could sum up the counts of letters and obtain the length of the segments. Here, they are totally independent parameters. You've got length of the segments, which is here. It doesn't have any connection to the counts here at all. That's for a segment. So you've got this for a segment. Then for the number of segments, you have to multiply you've got different factors k, so lambda start to depend on the k, and so I introduce this c at s will be the sum of the count, and n at s will be the length of the segment. So this will be the sum of the count of the segment. This will be the length of the segment. You've got two parameters now, and this product doesn't depend on lambda, and so you can obtain this kind of the function, and it is not normalized, but you can normalize it because it's again some kind of integral on lambda, and this integral may be taken. This is the likelihood. Again, we have to introduce some kind of a prior, which shows you the idea how often different lambda are found in this task. And you can marginalize using this kind of a prior and use this marginal likelihood as an objective function. What's important is that the constant weights do not work here. And this is the topic of conjugated prior in the field of Bayesian statistics. You can take integrals analytically if you select good priors, and in the case of Poisson likelihood, you have to select priors out of the gamma distribution. And if you consider this, you may have this kind of a marginal likelihood, and it's again the prior also two parametric. One parameter is related to the expectation of the length of the segment, and the other parameter is related to the expectation of the number of the counts within the segment. So you've got a bit more complicated factor here. So there are two parameters, alpha and beta, and beta characterizes the desired segmentation scale. So in this case, you even don't need to introduce regularization parameter. You can play with those parameters alpha and beta. So you can, by changing this beta parameter, you can select the desired segmentation scale. You've got enough parameters to, in your, prior distribution to get into this thing. So you found beta power alpha here. So it's kind of a regularization parameters which were introduced more or less artificially when we consider the Bernoulli case. So this is the example of segmentation at different parameters. So you've got some Poissonian data series, and you can see it gives you kind of a pretty noisy version for one parameter set, and you introduce a smaller beta, you got the kind of a more rough segment, more rough classes here. So it gives you, it shows you how it works. There are some more real examples. For instance, the ribosome footprints. And this is the ribosome profiling. Ribosome goes along RNA and make amino acid protein sequence, and you can stop ribosomes at different position, arrest them, and look on them. Look at them where they are stopped, because they're, in fact, they're moving with a different speed, and sometimes they wait for something, for instance, for the code, for the rare codon to come, and sometimes they're moving very fast. So you can see that there are different samples, and if they're shown with different colors here, so they are kind of replicates of the same experiment. And if you consider the initial noisy data, it's much more difficult to make inference here because you've got highly noisy data, and but there are some general properties of this. For instance, here the position where it goes very fast, there are no factors of reads which are coming from this region, and here they wait. So you've got this kind of a peak here, and in this case, some of them do wait and some of them don't. So if you consider the initial distributions of the cuts, it makes it very difficult to find real regions in the sequence. But if you do this kind of a segmentation, you can find that very often these positions of the segment are the same in the different data sets. And so this can be interpreted in biological terms. So it gives you real important observation because you make the correct approximation of your data, denoising, so you excluded the noise of the data set, as you can make much better analysis after if you consider the segmented data. Here the small set of references. So this is the cluster analysis, and this is the classic, more or less classic papers in which this thing has been published first. It's Olga value, and it's a very old paper of Scott and Knott, in which they introduced this kind of approach, I think, for the first time. It was somewhere in the 70s. And then I have several minutes to answer your questions because I have another meeting in 10 minutes. And then I give the platform to two of my students who will talk about a more complicated situation of studying copy number variation regions in the genome. So thank you for listening.